Well, welcome back to this next part of the colon cancer video. Now, today we're going to be looking at this paper here from the Journal of the American Medical Association. Rates of colorectal and other cancers are rising in young adults, as are other cancers. And we'll also be asking the question, is it plausible that mRNA vaccines are an etiological factor in colon cancer, particularly in cancers in general? We'll be looking at that in a little bit of detail. If you haven't got time to watch this video, the bottom line is, yes, I believe there are plausible mechanisms of action, which of course are an essential part of the Bradford Hill criteria. Anyway, let's look at what we've got specifically here. This is this paper here. Rates of colorectal and other cancers are rising in young adults, puzzling researchers. Just a little bit from this paper. Now, to be fair, this paper is from Journal of the American Medical Association, so it doesn't. Should we just say it doesn't speculate too much? But that's the link anyway. Incidence of 14 cancer types increased among younger people under the age of 50. So increasing in 14 cancer types in younger people. This, of course, is very concerning. 9 of those cancer types, including colorectal cancer and breast cancer, also increasing in some over 50s. So we're getting some cancers increasing in the under 50s, sadly, and some of those also increasing in the over 50s. There are some cancers, actually, where the incidence is going down. So there's a shift here. Quite what is happening. We're hoping to get down to some more of that in a minute. Factors, is it just increased diagnosis? No, I don't think so. Many early onset colorectal cancers are diagnosed after symptoms. So people are reporting symptomatic cancer. Now, in this paper here, and your son, Cancer Surveillance Research, American Cancer Society says this: given that similar trends have been observed in the US and many other countries, so this is a global phenomena around Western countries, it's crucial for researchers to address how much of this is driven by shifts in risk factors versus change in diagnostic practices. I think it's a change in diagnostic practices. I don't think it's diagnostic practices. We are diagnosing more, but I think there's something else going on in the etiology of this. Now, National Bowel Cancer Audit Group in the United Kingdom, 2024 report. That's it there. England and Wales, there was 35,779 patients diagnosed with bowel cancer. And this is just to show that between 2017 2018 there's more being diagnosed at an early stage, which of course is good, because 42% being diagnosed at an early stage means they're much more treatable. Now, interestingly, that's the 2024 report. The 2025 report didn't actually give the total number of cases. It shows a similar change in early diagnosis, but the number is actually 38,604 new patients. So to summarise, new diagnosis of colorectal cancer, England and Wales, 2021 to 2022, 35,779. 2022 to 2023, 38,604. So we do see, sadly, really quite a big, statistically significant increase in new diagnosis of bowel cancer in England and Wales. A little more detail just before we look at mRNA. By October 2020, monthly diagnoses had returned to pre-pandemic levels. So this is none of these effects that people talk about, that it's just lack of people not turning up for treatment during the pandemic. That's passed. From April 2021, the numbers of monthly diagnoses continue to rise. So from April 2021, so that's what, few months into the vaccine rollout, the numbers of monthly diagnoses continue to rise. Now, of course, this is a correlation. We're not saying it's causal. What we are going to look at, though, is plausible mechanisms. It is worth bearing in mind that in causality there will always be correlation, but correlation doesn't always equal causality, as we know. The total number of patients diagnosed with colorectal cancer during 2021-2022 surpassed the annual figures prior to the pandemic. Now, we don't know too much about these patients, unfortunately, from the UK data, but these are the number of patients. So Female, interestingly, yeah, male it was 56%, female 44%. Okay, I don't probably. I can't really think of a quick reason for that. Really, probably more men have smoked in the past. Maybe men drink more alcohol, but not a massive difference really between sexes. But the ages here, we are given this. Sadly, this is the 2024 data from the 2024 report. So age group under 50s, it was 6.4% of diagnosis. 50 to 59, 11.3% of diagnosis. These are the total number of cases. 60 to 74, we see the numbers going up, so they're the most common. We see that's the most common age range. Now, in the UK, actually gets slightly less common as you get older. Now, in the UK, we did used to start screening for bowel cancer at the age of 60. It's now down to 54. I think we screen between 54 and 75 now in the UK. It's just a simple poo test, basically, fecal occult blood sort of sample. It tests for chemical signatures of blood in the stool, which is a really good idea to do that. The 2025 report, so sadly, where here, what I was hoping to do is look at the increase in the under 50s. It was 6.4%. But the 2025 report, and this is from the National Bowel Cancer Audit in the UK, didn't give the number of under 50s. So this data is actually missing in the new report, which is really quite unfortunate. We really could do with this data updated for the latest figures, but we don't have it. It was missing from the 2025 report. So bit disappointing there from the report writers. Check it out for yourself. There's the references. Now, what I want to come on to now is it plausible that messenger ribonucleic acid vaccines are leading to some cases of cancers and bowel cancers in particular? So I've just picked up the criteria here. Causality may be adjudicated by larger scale associations, consistent between countries, where other explanations are unlikely, where the effect follows the cause, so it's cause followed by effect, where greater exposure causes more harm, with a plausible biological mechanism, with coherence between bench science and epidemiological data supported by even limited experimentation, by analogy to other causes of harm, and sometimes by reversibility. So it's very important we have the bench science in place, the plausible mechanisms. And here's some potential mechanisms via which mRNA could be leading to cancers. And these are currently described as theoretical mechanisms. First is DNA integration. Genetic information from mRNA vaccines getting into host, your DNA. Is this plausible? mRNA fragments get into the cells, of course. Then there's reverse transcriptase. So this is the enzyme that can convert mRNA into DNA. So normally in a cell, of course, there's the process of transcription where DNA becomes RNA, which makes the proteins. But the RNA can go back via reverse transcriptase and make DNA, changing the DNA of the host cell potentially. And if there's a change to the DNA of the host cell, what we call a change to the DNA in a cell is a mutation. So if that's happening, it's causing mutation. Of course, cancer is caused by mutation. So the mechanism there is plausible. Potentially disrupting tumour suppressor genes. So tumour suppressor genes are those which suppress excess cell division, or activating genes, oncogenes, increase the likelihood of cancers occurring. So it's quite reasonable to ask, well, I can see why we've got tumour suppressor genes, but why the heck have we got oncogenes? Why on earth should we have genes that seem to be designed to increase the likelihood of cancer? Well, of course, these are the genes that were active when we were fetus to produce the body, or when we were children to produce the body. It's just a problem when they go on working, because we want cell division when we need it, but then now in the adult form we don't really want too much cell division. If there's too much, that can lead to malignancy. to cancer. So it's plausible. DNA contamination integrated into the genome. I think this is plausible. I've talked to quite a few leading scientists who assure me that there are plausible mechanisms for this. The slam dunk information is not there at the moment. Can't think why it wouldn't be in the public domain. But the mechanism is plausible. That's the whole point. It's plausible. Contamination in vaccine products. So Pfizer, we know, contained modified RNA vaccine, and it also contained the SV40. That's actually a DNA contaminant. And the SV40 contaminant, we know this from talking to David Speaker, who did the primary work in Canada, who found SV40, simian virus 40 contamination. So the simian virus is the DNA that can inactivate tumour suppressor genes. So if you think about it, the tumour suppressor genes is normally suppressing the tumour. If you inactivate the tumour suppressor gene, for example, one called P53 and one called RB, if they're inactivated, then the tumours are not suppressed, so the tumours can go on and develop, promoting unchecked cell growth. So again, the theory makes sense. This is biologically plausible. This is not implausible. This is not ludicrous conspiracy theory. These are plausible biological mechanisms. Prolonged spike protein expression. Now, sadly, in the vaccines, they had this 1-methyl pseudouridine modified mRNA. So the uridine, of course, is one of the bases in the RNA, and the incorporation of this pseudouridine. So it's not the natural uridine that we would like. It's a false one. It's a biologically manipulated one, and that means the mRNA hangs around for longer. And we know that spike protein has been detected in patients after vaccination. From memory, we looked at one study where it was 700 days. To me, there's only two real mechanisms where the spike protein could be being produced for 700 days. One is the reverse transcriptase, that it's in our genes, which is probably the worst possible scenario. And the other is this pseudouridine allows the RNA to last for a long, long, long time inside our cells. Either way, spike protein can be being produced for long periods of time, which is not what we want. So rather a reckless experiment, in my view, to put that into the mRNA vaccines, but it was done and it's still there as far as I know. Encoding SARS coronavirus 2 spike protein, so the protein could be being produced over a long period of time. Another possibility is that lipid nanoparticles could be delivered to stem cells. Now, the stem cells are the cells from which other cells are produced. So you'll have a stem cell and that will divide and it will produce another stem cell. And the other one that divides goes on and produces a population of other cells. It could be anywhere. It could be in the liver. It could be in the skin. Could be the blood. So stem cells, if there's a genetic change in the stem cells, that can be passed on. Basically, it's basically a progenitor cell that can be passed on through many generations of cells onto future stem cells. And now we know that the mRNA lipid nanoparticle. The mRNA and the lipid nanoparticles goes everywhere around the body. We were deceived about that. We now know it goes everywhere. Of course, it's completely plausible that it could go to stem cells. It could go to any cell. Any cell. So completely plausible that it could go to, what do you call them? The stem cells. Sorry, yeah. Right, so sustained spike protein production will lead to chronic inflammation. Chronic inflammation, of course, can lead to cancer, disrupting cellular repair mechanisms leading to cancer. And this is kind of an axiom that, it's so well known that chronic inflammation leads to cancer. Chronic inflammation from human papillomavirus in the cervix. Chronic inflammation from gastroesophageal reflux disease in the esophagus. This is well known. Chronic inflammation in the colon from Crohn's or colitis, causing colon cancer. Another plausible mechanism, immune suppression or dysregulation, molecular mimicry. So it could be that some molecules on cells have a similar shape to the antibodies that are produced in response to the spike protein, therefore will start beating up on the body's own proteins. So if a vaccine stimulates a particular antibody, that antibody, yes, it can attack the protein which it was designed to attack, but it can also attack proteins on cell surfaces which look similar. It's called molecular mimicry. Sometimes they can be almost identical. And it basically starts beating up on our own tissues. This is autoimmune disease. And that leads to inflammation, and inflammation, as we know, can lead to cancer. Alteration of regulatory T cell function. So you get T suppressor cells and you get T helper cells. If they're dysregulated, that can potentially allow pre-existing cancers to progress. We know that T cell immunity, T lymphocyte immunity is totally vital. We learned this from Professor Dalgleish and Professor Clancy. And we know that Professor Dalgleish invented a simple bacterial preparation that can boost T cells that was submitted to the Medicines and Healthcare Products Regulatory Agency, who refused to license it, which is a pity because I would like it, because I would like my T cells upgraded. But I'm not allowed it. Neither are you. Outrageous that this isn't allowed to be prescribed compassionately after Professor Dalgleish went to all the bother of developing it and collecting data on it. Really is a major loss to humanity, and probably more specifically to me and you as well. Anyway, so that's again completely plausible mechanism. If the spike protein shares epitopes with tumor suppressor proteins, for example, so epitope is the part the immune system recognizes as being foreign. If they're on tumor suppressor proteins, those proteins will be beaten up by the antibodies. Therefore, the tumor suppressor proteins will be depressed themselves. Therefore, the tumor suppressor proteins will not be free to suppress tumors. So plausible biological mechanisms. Because of those biological mechanisms, we should plug that in, as it were, into the Bradford Hill idea, all these mechanisms. But of course, we don't have the national level data in most countries that allows us to compare vaccinated with unvaccinated people, or indeed the gradated risk with increased number of vaccines. So I will call for that data to be released so that a Bradford Hill analysis can be done, and then we'll know. But are there plausible biological mechanisms? I think in this video we've demonstrated that yes, there are plausible biological mechanisms that need further investigation. Can't imagine the British government's going to be paying for those. Can't imagine Big Pharma's going to be paying for those. So may not be done. We may never know until it becomes patently obvious in the future. And by that time, all the people that are making the decisions now will have been retired or died. The infected blood scandal in the UK only came out. It started in the 1980s, and the report was only last year. Was it last year or early this year? Last year, I think. So 30-year gap. So younger viewers will get the definitive information maybe in 20 or 30 years. Now, I hope I'm being over cynical, and we know before that. But so many things are covered up. Sadly, we might never know the truth, which, of course, in my view, is totally outrageous. There you go. We're just pawns in the game. More important people know what information we're allowed and what we're not allowed. We won't get mad. We'll leave it there. Thank you for